Okay, everyone. So it's time for us to go ahead and start with lecture 3. And so we're going to be focusing on the genetic material, and so DNA and how it's organized into chromosomes in eukaryotes, process of binary fission, mitosis, meiosis, and all sorts of other good stuff. So when you guys are ready, let's go ahead and get started. Okay, so what I'd like you to do right now is eventually pause the lecture in a minute or two and then think about, why do cells divide? Okay, so what's the purpose? Why might this be useful? Why do cells actually need to divide in our bodies as well as in any other cells? Okay, and then go ahead and stop lecture and then flip to the next slide. Oh, look, a green star. What does that mean again? Once again, you're only going to get that information from lecture, i.e., write this down. Okay, so remember that our cells will go through the process of mitosis, and we do this when we are growing, okay, and for development purposes. Also, when you scratch yourself, the way that your body repairs itself is through mitosis. Okay, so that's the process that's actually going on. In addition to that, when critters, not humans, okay, but when other critters who have the capacity to reproduce asexually, they do so through the process of mitosis. Okay, so 3 reasons that mitosis happens. All right, now meiosis, okay, that happens to produce gametes. So remember, those are the sex cells. The other thing I want you to consider, and we're going to go through this in some detail, okay, but I want you to be able to compare and contrast the process of mitosis with meiosis. So make sure you can do that. So start thinking about it now, and then we'll talk more about it later. How do you compare and contrast mitosis and meiosis? So what that means, or how are these processes similar, and how are they different? So the way our cells divide is based in 2 parts, okay? So the DNA has to replicate, and then it has to split and go into 2 different copies into the 2 different daughter cells, okay? And so the result of mitosis, you have the daughter cells that have the exact same DNA as the parent cell. That is really important. In mitosis, you start out with 1 cell and you end up with 2 that are identical, okay? If they're not, that's due to mutation or something along those lines, and usually a mistake. In meiosis, you start out with 1 cell and you end up with 4. And the key is that you have half the amount of DNA that you started with, okay? So what I want you to consider is, why is that so important? All right, well, it has to be that way or things would not work well. So for right now, think about, why is it meiosis you have to go down to half the amount of DNA? Why is that so crucial? Now, before we get into the process of mitosis and meiosis, with eukaryotes, by the way, which is the only place that those processes happen, let's step back to the prokaryotes. Okay, so think back. Pro means before, karyote means nucleus. So this would be the bacteria and archaea, which are the domains that we discussed on the first lecture of class. Yay! Isn't it great when the stuff, comes back full circle? So in bacteria and archaea, they have a single chromosome, and it is a circle. There's no nucleus, no other compartments, okay, no organelles, no membranes other than the one around the outside, okay. So that is their DNA. But they also have what's called bonus DNA, and that comes in the form of a plasmid. Now, what I want you to get from this is that in the chromosome, that has all of the instructions and information that the eukaryote needs to live. The plasmid, that's the extra stuff. So that might help it be able to glow in the dark, or it might be able to help the bacteria be resistant to antibiotics or something along those lines, okay? We end up manipulating plasmids, and that's how we, I don't want to say trick bacteria, but that's how we get bacteria to produce products for us, okay? Is by putting the genes in the plasmid, which, and an interesting side note, this just happened, it might be some trivia, if you're ever curious. How was the first critter modified through a GMO to do something for humans? It was actually when bacteria were modified to be able to produce insulin for humans. Okay, so next time you all have a biology trivia night, you can pull that little fun fact out. So you might be asking yourself, okay, so if the prokaryotes don't go through mitosis or meiosis, how in the world do they actually divide? And I would say, excellent question. They divide through the process of what's called binary fission. So what happens in binary fission is that the 1 copy of DNA, that is, the one that tells the bacteria how to live and function and so forth, reproduces to make another one. Then you get a newly synthesized copy of DNA. Then the cell grows and divides, does it again, grows and divides, does it again, grows and divides. Okay, that process is called binary fission. This is regulated by how many nutrients are around, what the conditions are like, all sorts of other good stuff. And I don't know about all of you, when I was in college, I used to have the really bad habit of ordering pizza late at night for dinner. And because I was up like cramming, studying, don't do that. Okay. And then I would let the pizza sit out overnight because we had too much left over to fit into a refrigerator. And I would have some, lukewarm pizza the next day for breakfast. Why is this important, you might ask? Well, what I do want you to know is that bacteria can actually replicate and, go from having one bacterial cell to two in a matter of 20 minutes, depending on the species. Okay. So every 20 minutes, they're doubling, and then they're doubling again, and then they're doubling again. So the next time you all leave pizza out, think about how many bacterial cycles it has gone through, how many generations, and how much you really want to risk the thought of food poisoning. Just a thought. So a nice way to actually draw this out in a diagram, which would be good for you to practice. And remember last time we discussed having total recall, where you pull out a blank piece of paper and write down everything you know. My suggestion to do that is with process binary fission, where you go from one bacterial cell to two. Okay, make sure you know the different stages. So you start out with the parent cell, the DNA replicates, then it splits into different sides of the cell. Next thing you know, you have daughter cells. Oh, look, another green star. Write this down. Okay, and once again, pull out that blank piece of paper and make sure by the end of lecture you can actually go ahead and recite all this fun information. All right, so the reason I want you to be able to understand how bacterial cells replicate and then split is because you should be able to contrast that with how eukaryotes do, okay? So now remember, a eukaryotic cell means it has a true nucleus. U means true, karyote means nucleus, okay? So what I want you to think about is, eukaryotes can be either unicellular or multicellular. They come in both forms. They have what are called cellular compartments. Okay, they usually have more than one chromosome. So what I want you to do now is think about, what does the nucleus contain? What does the mitochondria and chloroplast contain? What does each chromosome contain? Okay, make sure you can put down answers for these, and then go ahead and when you're done, go to the next slide. All right, so when it comes to the compartments of the cell, for example, you've got the nucleus, which has its own membrane, as does the mitochondria and chloroplast. So something that's really cool that we'll talk about later on is that the chloroplast and mitochondria at one point in time were actually independent living bacteria, okay? And early on, an early eukaryote ended up consuming one and then eventually consuming the other, would eventually turn into the mitochondria and the chloroplast, and that's called the endosymbiotic theory. Okay, hint, hint. Oh, look, another green star. The nucleus in eukaryotes contains linear DNA. How does that compare to the prokaryotes? You should know this. Okay. The mitochondria and chloroplast, on the other hand, contain circular DNA. Oh, put an A on the end of that. Okay. In addition to that, each chromosome contains many genes. That's the coding part of the DNA, plus non-coding regions. Initially, scientists thought that the non-coding regions were junk, but the reality is we're finding out that they might actually have some jobs that we didn't always think they had. So once again, make sure you write all this down and make sure it all makes sense. If not, let me know. All right, we're chugging right along, and the next thing we're going to talk about is the process of mitosis. So here's just a potential question for you. Prokaryotes go through the process of mitosis. No, you're not going to have this on an exam, but you should be able to answer this easy like piece of cake. Prokaryotes go through the process of mitosis as their cell divides. True or false? Make sure you can answer that. So let's talk about the eukaryotic cell cycle. Okay, so only, despite what we study, and despite what you may think, only a very small portion of the life cycle of a cell goes through either mitosis or meiosis. Okay, and remember, mitosis happens in the regular somatic body cells, meiosis only happens in the gametes. But what I want you to look is it's only a very small fraction. Okay, the rest of the cell's time is spent in either what's known as the G1 or gap phase, the synthesis phase, or the G2 or gap 2 phase. Okay, so make sure you know what part of the cell cycle, what part of the cell cycle that is spent on mitosis, okay? Basically what happens in G1, what I want you to realize is that it starts gearing up to replicate the DNA. Okay, the S phase, that's where actually the DNA is replicated. Okay, S stands for synthesis, all right? And then G2 is when the cell starts to gear up to go through the process of mitosis, all right? If you guys know that, you should be set. Now, during that very small fraction of time when the cell is spent in mitosis, these are the stages that happen, and you should know them, and you need to know what happens in them, okay? So during interphase, what's going on is the cell is, basically hanging out, okay? And so starting to get ready to go through the process of prophase. So during prophase, your chromosomes condense. Okay, so, and your chromosomes start to condense. Then during metaphase, meta means middle, they line up in the middle. All right, the anaphase, you can see they start to split. All right, they start going to each pole. And then telophase, that's when your cell starts to divide and split into two, or your nucleus does, I should say. Okay, so what I want you to be able to get from this, first of all, you got to know the steps. Okay, and I will add some supplementary information. Definitely check out the textbook on this section too. Okay, just so you're familiar with interphase, prophase, metaphase, anaphase, telophase, cytokinesis, all that good stuff, and what happens at each stage. All right, so the next thing I want us to be able to do is apply this information, okay? But first we need to go through a little terminology. So, once again, I'm going to train you all with the way I was trained, okay? Now, the way we treat what a chromosome is, because if you start talking chromosomes and molecules of DNA and all sorts of other stuff, it can get really confusing if you don't have a solid foundation. So, for this class, the way we're going to treat this is we call something a chromosome when it functions like a chromosome, and that will make more sense later, okay? So, once your DNA has replicated and gone through the synthesis phase, realistically speaking, you have two copies of it, but they're connected in the middle, okay? And that particular X functions like one chromosome. Hopefully that makes sense, okay? When it's connected in the middle and acts like an X, that's functioning as one chromosome. All right, but that one chromosome is made up of two sister chromatids, all right, or two sister molecules of DNA, and they should be identical. Knowing this piece of information, you should be able to easily look at this question and answer it. Which brings us to the answer of this question. Okay, so once again, how many chromosomes? So functioning as a chromosome, in this case, there's 3 X's. Okay, now how many molecules of DNA or sister chromatids that are identical are there? There are 6 total. Therefore, the answer is B. That doesn't make sense. Let me know ASAP. All right, so let's get into a little more detail about what happens at each phase. Okay, so during interphase, I mentioned it a little while ago. I said you could not see the chromosomes, and that is still very true. The genetic information is being accessed by the cell. Basically, it's doing the things it needs to function as a cell. Therefore, the DNA is not visible. Okay, so you can't actually see it. So just realize that, genetic information is being used during interphase. That's why you can't see it. And it's not until the next stage that you actually can see it. Okay, so other bits of useful information. Oh, look, a green star. So if you have an unreplicated chromosome, that is one double helix of DNA. Remember in the very first lecture, I believe it was, we talked about the story of how the DNA was actually, the structure was discovered because Watson and Crick, yada, 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 robbed from Rosalind Franklin. Okay, but they discovered it was a double helix of DNA that is one unreplicated chromosome. If you have a replicated chromosome, which happens during the synthesis phase, write that down, replication happens during synthesis, you will have 2 double helices of DNA. They're connected by the centromere, okay? When they're connected by the centromere, by the way, that functions as a chromosome. If it is one double helix of DNA, has not been replicated yet, that it functions as a chromosome, all right? The other thing I want us to start talking about are what are known as alleles. Okay, so alleles are forms of genes. Okay, so the alleles are what give us all of our charming characteristics. And what I want you to know is that we are diploid critters. This means we get half of our DNA from our mom and half of our DNA from our dad. But we only can potentially have 2 alleles in us. Okay, on a single chromosome, you'll have 1 allele. Once that replicates, it's going to be 2 copies of that allele, but they better be identical or there has been a mutation of some sort or problems. Okay, hopefully that makes sense. If not, definitely let me know. I do want to try and upload some additional videos that might be helpful too. All right, the S phase. This is when you have chromosome replication. Remember, S is synthesis phase. So you go from having 1 chromosome with the centromere that functions as a chromosome to go through the S phase, having 1 chromosome made up of 2 sister chromatids. Okay, the sister chromatids, by the way, should be identical, or once again, something's gone wrong. And so let's say, oh, look, a green star. You were asked to label these. Okay, before the S phase, it's an unreplicated single chromosome. After the S phase, it is a replicated chromosome composed of 2 identical sister chromatids. Write this down. This needs to make sense. Make sure you're okay with it. All right, so all the steps we talked about, the G1, the S phase, the G2. By the way, all that occurs during interphase. That's when the cell is hanging out, doing its thing. very small slice of DNA, or sorry, I'm tired. Very small slice of the life cycle, that is the M phase. And you might be asking yourself, what did she say the M phase does again? And I would say, oh, excellent question. Remember, this is in the somatic cells. Therefore, what's going on in the M phase, which is labeled M for a reason, would be mitosis. And remember, what goes on in the mitosis phase is that you have your replicated chromosomes and they separate into two distinct cells. All right, now what I want you to do is to take a minute, read through the question, okay, look at the choices, and then decide which one's the correct answer. All right, so once again, we're going to use chromosomes as when the definition of that is when something is functioning as a chromosome. So we're going through mitosis. You start out with 5. How many chromosomes will be present in the nucleus of each daughter cell? Not a trick question. The answer is right there. I know you guys know this. And once again, as you would expect, because in mitosis you start out with a certain number of chromosomes, you end up with the same number of chromosomes. So you start out with 5, you should end up with 5. Okay? So it's very important that you understand this, and once again, realizing that we call a chromosome based on when it functions like a chromosome. Oh, look, a blank slide. Perfect opportunity for you to take out a blank piece of paper. I want you to stop the lecture. I want you to start with a cell containing 2 unreplicated chromosomes, and I want you to draw out the process of mitosis, including every single stage that you label. Intend to be able to do this. All right, so this means that you start out with a cell with 2 unreplicated chromosomes. You take it through replication, take it through all the different processes that we've just been talking about. You should be able to do this by memory. My next suggestion to you is to then manipulate the number of chromosomes. Go from 2 to 4, 4 to 6, then go with an odd number. Doesn't make a difference. Okay, so make sure you can go through all the different examples. Make sure you feel comfortable with it. It's a great way to study. Here we have a nice drawing to reinforce what you've already put down, okay? Go ahead and label the different parts. Oh, look, interphase. Then there's prophase, metaphase, anaphase, telophase. Make sure you can label all the phases, and of course, make sure you know what's going on in each one. The other thing I want you to be able to do is to take a pair of homologous chromosomes, all right? So you might be asking yourself, what is a homologous chromosome? Well, remember I said a little while back that we are diploid critters. We have one set of DNA from our mom and one set of DNA from our dad, okay? So our chromosomes have a particular staining pattern that can be individual from other chromosomes, but that each one has a mate, okay, or has a pair. So chromosome pair number 1, chromosome pair number 2 has a particular staining pattern. It's based on the genes that those chromosomes hold. They're not identical. They can't be because we get half the set from mom and half the set from dad. However, they have similar genes on them for eye color, hair color, all those good things that give us our charming characteristics. What you need to know from this is the S phase. So you're starting out with a pair of homologous chromosomes. They go through the synthesis phase. Then you end up with homologous chromosomes that have been replicated. Each are made up of 2 sister chromatids. By the way, this is called a tetrad because there's 1, 2, 3, 4 copies of that chromosome. 2 are identical to one another, and the other 2 are identical to one another. Okay, so take notes all over the slide. And these are the notes I want you to be writing all over the slide. Okay, homologous chromosomes have the same genes but different alleles, and that should make sense. If they had the same alleles, we'd kind of be in trouble. All right, having too much, if you're too genetically identical, nothing good comes from that. So you have 2 homologous chromosomes with 2 different alleles, 2 different centromeres. One comes from mom, maternal, the other comes from dad, paternal. They go through the DNA replication, S phase, okay. Then you end up with 2 sister chromatids, same genes, same alleles that come from mom. Okay, 2 sister chromatids, same genes, same alleles that come from dad or the paternal. All right, hopefully all of this makes sense. By the way, what's a tetrad again? Make sure you know. Oh, here's an excellent application question. Assuming the arrows point to the same gene, do they point to the same allele or different alleles? Okay, so think about what stage of mitosis we're at. Think about where the centromere is located. Okay, think about how to address this question. Hopefully everything makes sense, and you chose A, the same allele, okay? So remember, it's 1 chromosome made up of 2 identical sister chromatids, and that's the way it's supposed to be. Therefore, that means that they have the same allele. Okay, now it's time for us to get into more terminology. Okay, so what is the difference between haploid and diploid cells? Now, if we were in class, I'd have you guys discussing, but of course we can't do that. So I would like you to take a moment and think about what you remember from basic bio that you tried to forget long ago. Okay, and think about what's the difference between a haploid cell and a diploid cell, and then write your answers here. Okay, so the difference between haploid and diploid cells, hopefully this is just a reminder. Oh, look, a green star, write this down. Haploid cells have one copy of each type of chromosome. Okay, diploid, di means two, means they have two copies of each type of chromosome. So remember, that's the maternal and paternal. Diploid cells are the product of fertilization following the process of sex, and we're talking the boring version here. So remember, the sperm is haploid, that connects with the egg, which is haploid, to form that diploid zygote, which will then grow up into a creature of some sort. So if you were to be shown a picture like this with a cell like this, and you were to be asked, do you think this is a haploid or diploid cell, and why? Okay, you should be able to look at this and think, okay, so how many copies of each type of chromosome do I see? How do I address this question? What looks like it might be the potential answer? Think about it a minute. It's not C. Okay, so you'll be able to know, but go ahead and choose your answer first and then move on. And with any luck, you picked haploid. You might be asking, how in the world do you know? Okay, well, there are 3 chromosomes here. Okay, and so it's an odd number. Additionally, well, there's 3 chromosomes. If it was diploid, well, it depends on what's going on. If it was diploid, okay, then you're going to have 2 copies of everything, so it's not going to be an odd number of chromosomes, all right? So this is not meant to trick you. I just want you to start thinking along these lines, okay, so you can start recognizing these patterns. All right, so once again, we have our homologous chromosomes that are paired up. Okay, they go through the process of replication. Then you have your homologous chromosomes after replication, which is known as a what? That's right, tetrad. Okay, so each part of the tetrad, you have 1 chromosome made up of 2 identical sister chromatids. The second chromosome, potentially paternal or maternal, made up of the identical sister chromatids. Can you tell I want you to be able to label and diagram these things? Now what I want you to be able to do is take allele A1 and A2 and draw them out on here. Hint, hint. Okay, take a moment and do that for a second. Okay, so now let's think through this. So we have our eukaryotic cell division, otherwise known as the process of mitosis, okay? You have multiple chromosomes. We've only been looking at 1 or 2 chromosomes, but the reality is humans, by the way, have 23 pairs of chromosomes. Okay, so think through that. What we have here is known as a karyotype. A karyotype is nothing more than all the chromosomes pulled out and stained up. But what you can see is chromosome pair number 1 here, chromosome pair number 2 here, chromosome pair number 3 here. It looks like they're connected, okay? And I'm sorry about that. That's just the way the karyotype came out. But the reality is, you know, that number 1 way up in the upper left is made up of 2 different chromosomes, 1 from mom, 1 from dad. Okay. Each chromosome, you know, has many genes, has 1 allele per gene, is made up of single double-stranded DNA molecule before the S phase. So this is before the S phase. Okay. Given this information, how many total alleles per gene do you have in a diploid cell during the process of G1? Okay, be able to answer this. Also, before you go on to the next slide, be able to address human somatic cells are what? Haploid or diploid? You should be able to answer this too. So the answer to the question, how many alleles per gene? Of course, 2. Diploid, di means 2. Okay, human cells are what? They are diploid. Okay, except the gametes, the sperm and the egg, are haploid. And they have to be, because otherwise when they come together, we'll have too many copies of DNA, and nothing goes well for humans when that happens. All right, so what we have here is a nice little diagram that has terms and definitions and even shows some examples for some things. What I want you to do at the very least is read through this. Okay, the other thing I want you to do is to learn it. If you ever plan to take a genetics class, this particular table should be second nature to you. Okay, and if it isn't, then I've not done my job. Have I said you want to be able to know these terms? Okay, hint, hint, know the terms, take your time, read through them, all right, and be familiar with them. Okay, so as we're chugging along, it's time for us to dive into meiosis. So I am telling you now, a question I am going to put on the exam is to have you compare and contrast the process of mitosis and meiosis. It's not going to necessarily appear in that form verbatim, but have I mentioned I'm going to ask you to be able to do it? So as we go through the steps of meiosis, be able to think back to mitosis, okay, and be like, well, I wonder how that compares to that other process we talked about earlier in lecture today. Now, let's think back to that first lecture on day 1, okay? And we talked about what does it mean to be alive, and we talked about what properties do you have when you're considered alive. And if you will recall, one of them happens to be able to reproduce. And again, we're sticking with the boring version, okay? So critters can reproduce multiple ways. It depends on the species. Some of them still stick with the method of asexual reproduction. Okay, I have a spider plant that has all sorts of little spider plants that are genetically identical, to the initial spider plant, which by the way is like 30 years old now. Whew, I'm getting old. Anyway, so other critters, on the other hand, reproduce sexually. Okay, and again, we're sticking with the boring version. However, the thing I want you to think about, what advantages and disadvantages might you have to each of these methods? And again, boring version, guys, okay? From an evolutionary long-term perspective, what might be some advantages and disadvantages of asexual reproduction versus sexual reproduction? And again, keep it boring. If you know what I mean. All right, so as we're considering asexual reproduction and sexual reproduction, okay, asexual, you start out with one parent and they produce genetically identical offspring. Lots of plants can do this. Bacteria do this. That's how they do that, through fission, okay? There are a very small number of animals that do this, even one vertebrate lizard that can do it, okay? Now, sexual reproduction is the flip side, where you have two individuals that combine genetic information and they produce offspring that are genetically different. Why is that, from an evolutionary perspective, going to be an advantage? All right, so I want you to think about that now. Why long-term evolutionary is genetic variation a good thing? Now, hopefully you've downloaded the slides from class, the PowerPoint slides that are, again, they're in the module section. And you will see this slide. And what I want you to do are write the advantages and disadvantages of sexual and asexual reproduction. Once again, from the evolutionary long-term perspective, what might be some advantages and what might be some disadvantages. Now, if I were asked to fill out this table, it might look something like this. So, sexual reproduction advantages, evolutionary long-term perspective, we're talking more genetic variability. You'd be able to adapt to new environments. Have genetic recombination. So if you have bad mutations, they're going to get purged from your population. Realistically, though, disadvantage, it costs energy. I've been married for 20 years. Oy. Anyway, so when it comes to, partnering up and so forth, the disadvantages, courtship time. It takes a lot of energy and resources. Again, married for 20 years. So additionally, a disadvantage is only females will produce, the new offspring. Okay, so then let's think about the asexual reproduction. Well, advantages, it saves energy. Don't need a courtship, don't need a mate. Okay, faster population growth. But from a long-term perspective, realistically speaking, low genetic variability, so you can't adapt to new environments. It gets really hard unless you have some somatic mutations that happen, and those generally take a long time. And realistically, the only way to eliminate bad mutations is death, okay? So, which sounds kind of sad. All right, so meiosis. This is the process of that is required for sexual reproduction. And then, of course, fertilization. So the whole idea is meiosis produces your germ cells or your gametes. And remember, they have half the number of chromosomes and half the amount of genetic information as the original cell. The good thing about this is it increases genetic variation, all right, and therefore the more genetic variation you have, your population can actually evolve if need be, and new conditions are happening. All right, so what I want you to do now is stop the lecture again, pull out the slide and the PowerPoints, and fill this out based on what you feel is, the most important part of each of these, okay? So address these questions, all right? Do the best of your abilities, and then when you're ready, start the lecture up again and go to the next slide. So if I were asked to answer these questions, all right, the end result, you get your gametes, your egg and your sperm, that are genetically different from the parent cell. Additionally, they have half the genetic information. How many rounds of cell division occur? 2. You got meiosis 1 and meiosis 2. How many daughter cells are produced? 4 daughter cells, and they are haploid, okay, meaning they got one copy of the chromosomes. What type of cells undergo meiosis and where are they located? They're the germ cells and the sex cells, and they're located either in the testes or in the ovaries. Hopefully that all makes sense. All right, so meiosis produces haploid cells that have half as many chromosomes as the parental diploid cell. Okay, so you've got, the egg and the sperm come together on this cycle. You get the fertilized egg, which then goes through mitosis to grow up to the full adult. In this case, we have a puppy. And so then in animals, in the ovaries and testes, they will go through the process of meiosis to produce the eggs or the sperm. Those get together, and the cycle starts all over again. This should all make sense. So dogs, we know, are diploid critters. They have 78 chromosomes. Therefore, in their eggs and their sperm, they're going to have half that number, which is 39. Okay, and by the way, haploid is represented by an N. All right, diploid is represented by a 2N. Then those come together to form the zygote. 2N equals 78 again, and you can trace the cycle as it starts all over again. So remember, once again, meiosis occurs in diploid cells that have pairs of homologous chromosomes. So what I want you to think about are what are the characteristics of those homologous chromosomes. All right, take a moment, think about it, and then move to the next slide. Oh, look, here we have a nice list. Okay, so homologous chromosomes are similar in size and shape. They contain the same genes in the same order. That makes a difference, by the way. They have similar DNA sequences, but they're not the same, or at least they better not be. And then they can carry different alleles of the same gene. So the way to think about it is a pair of shoes. Not completely identical, but still similar enough, having the same characteristics when it's actually important. Okay, so once again, you have your homologous chromosomes with your two different alleles, big B, little b. Goes through the process of DNA replication. At what stage of the cell life cycle, by the way? You should know this. Then that creates the homologous chromosomes, okay, and the tetrad where you've got the sister chromatids, and we have a new term to introduce, the non-sister chromatids, okay? So sister means they're identical, non-sister means they're not, however, they still have the same genes on them, and that's important. Okay, so once again, I want you to draw out a diagram. Oh, look, I got a blank paper. You've got a diploid germ cell, 2N equals 2, you should know what this means. 2N is diploid, 2 means 2 chromosomes, okay? Go through the process of meiosis and be able to draw out each step. Label them too. Okay, so my suggestion to you is to always draw with 2 separate colors to be able to distinguish maternal from paternal. Hopefully you do something that looked a little bit like this, okay? In which case, what I want you to realize is that each of the stages are here. Meiosis 1, by the way, is known as reductional division, okay? Because ultimately, you're reducing the number of functional chromosomes you have in each cell. All right, and then the second meiosis 2, the second stage, is known as equational division. All right, so you start out with a certain number of chromosomes and you end up with the same number of chromosomes. Okay, so hopefully that makes sense. Okay, so given what we've talked about so far, this question in blue should be a piece of cake. The 2-way chromosomes are what? Think about what we've said. Think about what I suggested you about 2 slides ago. Actually, 1 slide ago. This should be a piece of cake. All right, if you answered B, chromosomes from the same parent, yay, that would make me so happy. Okay, so that is the correct answer, and that's what those particular colors are meant to indicate. Now, from an evolutionary perspective, genetic variation is crucial to the long-term survival of a species. So, anything that gives you high genetic variability from an evolutionary perspective is going to be preferred to a process that gives you low genetic variability. Okay, so something to definitely keep in the back of your mind. All right, so the way this genetic variation is introduced and the way it happens, there's 3 different ways. And so what we're going to do now is talk about the first way. So you've got these arms of the chromosomes that are dangling. They bump into one another. They exchange arms, okay? In which case, that location is called the chiasmata. It's where they're held together, then they break apart and exchange arms. That is when you get recombination. And so what I want you to consider then is if you were to follow these chromosomes through meiosis, you would end up then with 4 different, daughter cells that all have different genetic information rather than just 2 and 2. Okay, so the end result is increased genetic variation from an evolutionary perspective. That's good. This is the first way that, having maternal and paternal chromosomes recombine during meiosis, that you get this variation. So as I said, it creates unique chromosomes. This gives you unique combinations potentially of different alleles of genes, and ultimately that's a good thing from an evolutionary perspective. Write this down. Okay, so the second way that you introduce genetic variation, by the way, was discovered by Gregor Mendel. And this is the law of independent assortment. Okay, and what this says is how your chromosomes line up with regards to maternal and paternal is random. So, for example, if you have a cell with only two chromosomes, you have half a chance of having probability 1, where mom chromosomes are all lined up and dad's chromosomes are all lined up. Okay, and then follow that through and think about how it goes through meiosis and what you end up with. Follow this through. Okay. Now, the second probability is you get them mixed. Okay, so you get them rearranging themselves. What this means is that how one set of chromosomes assorts itself on the equator is completely independent of how the other sets of chromosomes assort themselves on the equator. That's why it's called the law of independent assortment. Okay. Now, if you were asked to actually calculate out how many different combinations it could be, well, it could be a lot. So in the formula for that, by the way, is 2 to the nth, and n is the number of haploid sets of chromosomes. Okay, so humans, we've said, have 23 haploid sets of chromosomes. Therefore, we have 2 to the 23rd, or 8 million different potential combinations of chromosomes. That's why when you have siblings, you don't look exactly like them unless you're identical, okay? Because there's all these different possibilities of how these chromosomes align up on the equator. Once again, evolutionary perspective, a good thing. So just to add some labeling, 50% of your germ cells, if you have two chromosomes, will line up like on the left. 50% of your germ cells, if you have two chromosomes, will line up like on the right. And because of that, you get unique combinations of genes, i.e. genetic variation. Now, last but not least, the third way that sexual reproduction introduces genetic variation is that it's a completely random process as far as which egg and which sperm will come together to form that zygote, okay? So your gametes are going to combine randomly at fertilization. So think about all of the different possibilities, okay? All the different ways that you could have these different combinations coming together. It's potentially 70 trillion, okay, different combinations. Once again, that's why you're not identical to your siblings, but you do share some of the same genes. So what does this mean? The third way we get increased genetic variation is through random fertilization, random pairing of, the male and female gametes gives you unique zygotes and unique, embryos, children, and so forth. All right, everyone, it's time for us to apply this again. How much of your genome comes from each of your parents? We've said this before. Keep it simple. Sorry. It's not E, by the way. Okay, so take a moment and address this question. All right, so if you guessed B, we would be doing a small happy dance in class because that's the correct answer. Because remember, because of meiosis, each gamete contains 50% of your parent's genome. Therefore, it's split equally between your two parents because, again, we're diploid critters. All right, so once again, comparing the process of mitosis and meiosis. Mitosis, you get two genetically identical daughter cells, only happens in eukaryotes and only happens in somatic cells, in other words, the body cells, okay, not the gametes. Meiosis produces four daughter cells. They're each genetically different. Once again, only happens in eukaryotes, but only happens in the germ cells. This hopefully makes sense. If not, you need to come find me. All right, I've said this before and I'll say it again. You need to be able to take this slide, preferably only look at the left to start with, and be able to draw and take it through the process of mitosis, and then draw and take it through the process of meiosis, okay? Start now with two chromosomes. Make sure that you can go ahead and draw it and trace it through, label each stage, and be able to briefly say what's happening at each stage. Also, label your tetrads, label your chromosomes, label your centromeres, that's the middle where they attach, okay? Label all the other good stuff, and be able to tell me where the genetic variation happens in meiosis. Nice little table to be able to summarize and compare and contrast the differences and similarities between mitosis and meiosis. Okay, so go through and address each of these questions and determine whether or not they happen in mitosis or meiosis, and go ahead and fill it out. Okay, so it's time for us to have some additional questions to test your understanding and your knowledge. So my suggestion to you, again, pull up the slide and then stop the lecture, and based on what you've learned today, go through and try to answer the questions, okay? And then whatever you don't get correct, then need to do a little more research or need to contact me and let me see if I can help clarify some things for you. Okay, so this is your first question, okay? So stop for a moment, consider what the ploidy is of the cells, and see what your answer is. It's not E. Hopefully you chose haploid, okay? Because obviously in the very beginning it was a diploid cell. Remember we said meiosis 1 is reductional division, so you lose half of your genetic material, okay? And then the second step is known as equational division. That's going to come in handy in a minute, but just remember that for right now. Meiosis 1, reductional division. Meiosis 2, equational or equal division. So now we're going through meiosis 2. Starting out with a certain number of chromosomes. How many chromosomes do you end up with when you're done? Once again, stop the lecture, address this question honestly, and then move forward. Hopefully this is what you guys came up with. You start out haploid, 2 chromosomes, you end up haploid, 2 chromosomes. Equational or equal division, okay? So once again, let's try this question, okay? You have salamander species A, which has 60 chromosomes and they're diploid. You have salamander species B, which diploid, and 98 chromosomes. 2 individuals of species A mate with one another. 2 individuals of species B mate with one another. Assuming no crossing over, who's going to have more genetic variation among the offspring? Okay, it's not D. All right, so, and think through the question, answer honestly. You guys should have this, should be a piece of cake, and then move forward. With any luck, you chose B, species B. You might be asking yourself why. If you didn't, it's because species B has more chromosomes, okay? The reason you're assuming no crossing over is because in theory, species A could have more crossovers than species B, and we wanted to keep it simple and straightforward. So this is only based on the number of chromosomes, in which case, remember, 2n equals 98, so they have 98 chromosomes. 2 to the nth is going to be 98, way more possibility for genetic variation there than in the first species. Okay, so this question is meant to push you a little bit, okay? So the first thing that we do not do is panic when we see a word problem, okay? So whatever, habits you might have had from high school, it's going to be fine. No panicking allowed. Secondly, read the question carefully, okay? And then write down any useful information that you might need. Then what I want you to do is go through and address the question. I just realized something got lost in translation. The first choice is A, second is B, the next one is C, then D, and then E. I don't know what happened with that, okay? Either way, go ahead and work through, and it doesn't matter the letter, but circle the appropriate choice. How many chromosomes, then how many chromatids, and so forth. So follow through, read the question, and once you're ready, then move forward. Okay, so if all went well, you chose what would be labeled properly had it worked as choice C, okay? And that's the correct answer. So let's think about this. At each one of these, by the way, it should be at the beginning of the stage of metaphase 1, at the beginning of metaphase 2, and then you read the next one. So 2n equals 50. Okay, so how many chromatids, sister chromatids, do they have at the beginning of metaphase 1? Sister chromatids, double it, it should be 100. Hopefully that makes sense. Then, how many chromatids would there be at metaphase 2? By then you've gone through reductional division, so you should have half. Half of 100 is 50. Okay, then towards, and again, it's at that point in time, it's still sister chromatids. Then, how many chromosomes would there be in each egg cell? Okay, at that point in time, so remember, it's the beginning of metaphase 2, so it hasn't gone through the second split, and it's talking about chromatids, okay? And so at that point, the X's are still together. But by the end, okay, how many chromosomes do you have in each egg cell? Well, remember, it's functioning chromosomes, so that goes down to 25. And that should make sense, because remember, in the very beginning, we started with 2n equals 50. So by the end, it's n equals 25. Don't let the terminology throw you. I'm just trying to have you apply things and turn it around in your head so you become much more familiar with it. And again, if you have any questions, let me know. That will be the end of lecture for today. I wish you all a wonderful weekend. If you have any questions, of course, you guys know how to find me. And I will talk to you next week. Stay safe and healthy, everyone.